I'm not going to really go too much into like really nitty-gritty of bioinformatics. The section is supposed to be about bioinformatics for CRISPRs. I'm really going to try to focus on practical application. I'm going to talk about what types of servers, for example, are out there that you might use. How do they work very critically? You don't want to treat these things as a black box. And I'm going to focus a little bit on some of the questions that people might have for using these. And to try to really kind of demystify the whole thing. I mean, it's called bioinformatics, and sometimes people who are not bioinformaticians see that and they get a little bit scared. Bioinformatics is really just stuff that you would do yourself, that you could write down on a piece of paper. It's just done really, really fast, right? It's a person that thinks of these things and then codifies that into something that a computer can do at millions of operations per second. There's no magic. It's especially in the case of CRISPRs, finding guides, things like that, tends not to be rocket science. And I'll tell you a little bit about how that works. So let's start off with one of the central problems that if any of you guys have tried to do a cutting experiment or an inhibition experiment or anything like that, this is one of the central problems that the field is currently facing right now. So here are a couple of guides, and these are going to be for SPI-Cas9, Pyogenes. They've got NGGs at the end. And just from looking at these, does anybody have any clue, like what makes these different, right? Are these different? Are they not different? Are they basically the same, right? GC content. OK, that's one thing. OK, so which one works and which one doesn't? Which one's good, which one's bad, right? This is kind of the challenge, right? Some of the GC content is through the roof, and it's really crazy. But this is EMX1. This is one of the most highly active guides that people have found so far. It seems to be relatively specific. It's great. This guy, great, has high GC content. This is VEGF site 2. It's very, very highly active. But when people go looking for this in sequencing experiments, which I think Dana has talked about already, this thing hits tons and tons and tons of genes. Okay? So we've already identified one thing that might be bad about guides, just from the sequence alone. And this is a guide that we've tried in the lab. And I challenge you to find something different between this guide and this guide. This guide is completely inactive. It does absolutely nothing at the target. And this one is very highly active. And so at the moment, the state of the field is kind of, we're trying to come up with rules and figure out what differentiates these guys from each other. What makes one guide active and what makes one guide completely inactive. So any kind of guide finding algorithm, whether you're doing this in your head or you're doing it on a computer or anything like that, comes down to this. You want to first find the guides. So basically, where are places that would be guides? You want to map them to some kind of reference genome. And this tells you both where the guide itself is, and it also tells you potential off-targets, because maybe it maps to 20 different places. OK, that would be bad. Or is this a very unique sequence in the human genome? And then you also want to know other things about the reference genome. If you have a whole bunch of potential off-targets, are there genes in those places? Are they gene deserts? Are there other things that we know about it? Maybe there are DNase hypersensitive sites that might indicate some sort of chromosomal packaging. And this is sort of an ongoing area in CRISPR bioinformatics. And I'm not going to talk too much about it, because at this point, that data is sort of in progress. We don't really know a lot about how chromatin state affects the ability to induce cuts or inhibit or things like that. It's sort of anecdotal at this point. And then finally, you want to, once you have a guide and you've incorporated all this stuff, you want to score it. Is this a good guide? Do I think this is going to be a bad guide? And that incorporates features about the guide itself. And it also incorporates things about those annotations. So again, if you have maybe 3 off-targets, but every single one of them is in an essential gene, that might be really bad if you wanted to do some sort of survival screen. So what I'm going to talk a little bit more is some of the stuff about features of the guide itself, how we incorporate that into off-targets, things like that. First, we'll start right at the beginning, and we're going to talk about finding guides. Just to let you know, we're going to go through sort of a little bit of this whole thing in detail for 2 of the most popular servers, and try to sort of peel open the box and show you a little bit about how these things work, because it's not totally intuitive. All right, so how do we find guides? Let's say we've got this sequence. We want to look for PAMs. Okay? So let's say we're working with SPI-Cas9. Where are the PAMs in this? It's a short sequence, so I think it probably wouldn't be useful to have people in the audience point. But I think the most obvious thing is people always find the forward PAMs first, right? You look for NGG, and you see those, and you're like, oh, great, they're all my PAMs. But always remember, CCA is a PAM on the opposite strand. So anytime anything is finding guides, it's always looking for CC something, okay? CCN. And also don't forget that there can be overlapping guides. And then all you do is you just count backwards by 20 bases. And those are all of your potential guides. Okay? So This is a potential protospacer, so is this. And here's the one on the opposite strand. You just reverse complement it, and then that's your protospacer for the other strand, right? Whenever you have any kind of web server that's finding guides and spitting out huge lists of guides, that's all it's doing is just a text search for something GG in the case of SpyCas9 and spitting out whatever bases were behind it, okay? So now we've got this huge list. Let's say we had an entire gene. So we've got hundreds and hundreds and hundreds of these things. And now we have an entire sea of a genome, and we want to figure out where are potential off targets, where does the protospacer itself match, is this in a gene, all that stuff. So how do we find out where the protospacer is, or sequences that are like it? Okay, so this, I'll just open that up to you guys for the moment. Okay, this is a problem that has been solved before in bioinformatics, and you've heard a little bit about it in terms of other revolutions in biology. So let's say we have an entire genome, and we want to figure out where I'll give you a hint, it's a short piece of DNA that you have read in an experiment. How do you figure out where that goes in a genome? What does this sound like? You've got a 20 mer, or maybe it's 150 mer. So BLAST works really well if you have if you've got an entire gene, you want to find homologs, things like that. What's that? Primer search can do it, yep. But a really, really fast way. What's that? Tag scan, yeah, tag scan can do it. But this is really similar to resequencing approaches, right? So if you want to find SNPs, if you want to reassemble genomes, all that kind of stuff, this is exactly how people take short reads from HiSeq and MiSeq experiments, and they can just map them together. So just pretend that a protospacer is a read from a sequencing experiment. And programs like Bowtie is not the only one. There are others. These things can map hundreds and thousands and millions of reads in a matter of minutes to hours. Okay? So taking a 20 mer and saying, where does this occur in the genome is like nothing for programs like Bowtie. This even allows things like fuzzy alignment. So you can say, I've made this quote unquote read, but I don't know exactly what the sequence is because of sequencing error, and so maybe these bases are different. And so that should be raising in your mind, oh, this sounds a little bit like maybe off-target matching, right? These bases matter, these other bases don't really matter, and so I'm looking for something that is strict in this region and not so strict in this region. So a lot of times when you see people talking about they mapped protospacers or they looked for off-targets, things like that, a lot of these pieces of software are built on Bowtie. So let's say we know where the guides are, okay? So these are just chromosome coordinates. We figured out, well, this one's on the minus strand here, and this one's on the plus strand over here. So what do we do with that? We need to figure out what's at those places. And I'm not really going to get very much into the detail of this because there's about 1 billion different ways to do this and 1 billion different databases. But if any of you guys have ever gone to the UCSD genome browser or the Ensemble genome browser, and you've seen these massive tracks of data with encode databases, methylation state, all that sort of stuff. That's all that this sort of stuff is doing. You have a given locus, and then you go into the databases and you say, what do we know about that locus? What else is there? So this is what these look like graphically. So you might say, oh, my guide anneal's right there. Is that in a gene? Is it not in a gene? Et cetera, et cetera. So when you go to a web server and it tells you, here's a gene that's at this place, here's a transcript at that place, all it's doing is querying to some database. It's pretty important to know what's in those databases. This is one of those cases where it's very tempting to sort of abstract in your head. It tells me this thing is here, and great, now I know. I'm not really going to get into the details of this, but these databases are unfortunately not identical. So UCSC is really geared for one thing, Ensemble is geared for another thing. They contain sort of non-overlapping sets of data. The really key thing is to know what your server of interest is doing and make sure that it is doing what you want it to be doing and that it's using a data set that you're interested in. So we'll talk a little bit about that for one server. For example, the CRISPR.MIT server uses UCSC, which is great for so-called reference genes, but does not hold a huge amount of transcripts. Ensemble holds a huge amount of transcripts. So if you wanted to do, say, a CRISPRi experiment, it might be not the appropriate database to use. Okay, so great. Now we know where guides are. What do we want these guides to do? Okay. Well, the first thing we want for a guide is we want it to be active. Before we get into any of the bioinformatics, I'm going to talk a little bit about some of the things that are sometimes but not always taken into account by these servers. They're easy to forget, but they're also very easy to avoid. So these are things that typically are things you would do yourself. So first off, most guides these days are expressed from Pol3 promoters. And when you're using Pol3, if you have 4 T's, that is a U in a row, that's going to terminate the guide. Never, ever, 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 ever choose a guide that contains 4 T's in a row. Almost none of the servers that are out there will tell you this. They'll propose those guides just fine. They don't triage them. Don't make them. Your guides will never work because you're never going to make the RNA, okay? Unless you're going from a Pol2 promoter or you're doing something kind of non-standard. But if you're using any of the normal guide expression constructs, I would avoid these. Avoid homopolymers in general. As you saw from GC content, it's very easy to make guides that contain 5 C's in a row, stuff like that. There is data out there that these are usually not very good, but there's no real agreed upon scoring metric to say this is bad. People have started to implement this kind of stuff, but it really ends up being kind of a heuristic, like, this guide is predicted to be good, but then post hoc we see it has all these homopolymers, so we downweight it. GC content has started to emerge. You guys pick this up right away. In general, avoid guides that have very low or very high GC content. The range that you can go for is very big. This is pretty easy to calculate. There's really no reason to make a 90% GC guide or a 5% GC guide. This is something that you also heard a little bit, I think, from Max. You want to, and maybe Jonathan too, you want to avoid stable secondary structure. There are a couple of places that do incorporate these kinds of scoring metrics. At the IGI, we have some tools that I'll tell you about a little bit that incorporate this. In general, you want to have 2 things that you avoid. And just in terms of predicting secondary structure, by the way, you can predict RNA secondary structure with servers like Vienna. So this is not something you have to install or anything. You can just go and check this out. You want to avoid 2 things. First off, if you have a protospacer, you want to avoid very, very stable secondary structure in the protospacer. Okay? So how stable is very stable? There's not really good rules for that yet. But if you take just the protospacer sequence and you predict secondary structure and it's like a perfect hairpin with minus 15 kilocalories per mole, the chance of that being able to unzip and anneal to your sequence of interest, it's probably going to be fighting an uphill battle. The other thing you want to watch out for is if you have a protospacer, maybe it doesn't have secondary structure in itself, but it can very easily pair back with a constant region, that could disrupt activity as well. So these things are, again, not typically taken into account in published servers and published methods, but I am aware of a couple of sort of in-process things where people are incorporating this. And this is one of the things that we do at the IGI as well. Okay, so these are the rules of thumb, but surely it's more complicated than that. You guys sitting in the audience, if you saw a perfect hairpin, you could probably just see that in an RNA. Just by looking at a protospacer, you would see an inverted repeat. So, okay, we sort of went through all of that obvious stuff, and still some guides work really well and some guides don't. So maybe it is that there's something more complicated. So to good guides have certain sequences that bad guides don't. And so this is where we're going to get into a relatively new tool, which is from David Root's lab. This is where they try to develop machine-learned rules for what makes a good guide. All right, so you heard a little bit from Max about this. Let's say that we wanted to find out what makes a good guide. So we're given, in this case, a gene or maybe a whole bunch of genes, and we're going to do a cutting experiment. So what could we do to find out which guides work to disrupt that gene, which guides don't? So we got just one gene. It's not too big. Tile, perfect, exactly. So that's exactly what you do. You just take every single guide in the entire gene, and you say, does this work? Does it kill this gene? Okay. In this case, they choose cell surface markers, so it's a really easy FACS experiment. So all you do is you make every single guide, do a lentiviral library like you heard Max talking about, and you sort for loss of that protein and sequence those guides. That's where you get plots like this. If you have guides that go away very easily, that means they're good. You've disrupted the gene. But then there are a bunch of guides which happen to be in exons, which nothing really happens. Okay? There's one complication. David's lab did this in a couple of different cell lines, and they saw sort of OK correlation, but maybe sort of not OK correlation. So they did this in 3 different lines. And you can see that at the extremes, For guides that are really good or really bad, everything kind of clusters together, but in between, it differs between cell lines. So guide activity can certainly be cell line dependent. In general, it seems like if a guide is really good, it's going to be really good in most places. If a guide is a dog, it's a dog everywhere. Okay? The question is, what do we do about the stuff in between? Because all of these guys in between are, they work pretty well in one cell line. Maybe they don't work okay in the other line, but they seem to work pretty well. But how do you know about your cell line? That's kind of tough. Local sequence effects could be a problem, like things, SNPs in the cell line of interest, stuff like that. We don't really know. So now the question is, what about on average? Okay? What about, instead of looking at an individual gene in one cell line, we look across many, many genes in many, many cell lines? And so how would we do that? Well, the way you can do that is you take all of those enriched guides, and you basically just pile them on top of each other. And you ask, what are the sequence preferences of those places? And you do it across every cell line that you've looked at. And you try to hope that all of these specific things even out. Okay? And from that, there are actually a couple of really interesting trends that pop out. So first off, the most obvious thing that they pointed out was that if you have a G next to the PAM, this now here on the y-axis, these are things that are favored. Down on the bottom, those are things that are disfavored. So if you have a G at this position next to the PAM, that's really good. If you have a C down here, that seems to be pretty bad. And that seems to happen across a lot of different targets, a lot of different places where these map, and a lot of different cell lines. And there are a couple of other interesting trends. So for example, having a C here is not very good. Having a G here is not very good. A's seem to be preferred right here. But it seems to be kind of complicated, right? This is sort of looking at each position individually. And it's sort of hard to turn this into something that we can hold in our brain, or it's hard to turn into something that we can use in some sort of tool way. So what we really need is a predictor. So the way you do this computationally is you take each position, and then you take each base at that position, and you call it something, you call it a feature. Okay? And the feature basically just says, well, if I've got A at this position, is that bad or good? If I have G at this same position, is that bad or good? If I have T at this position, is it bad or good? Okay? And they do this in a very binary way. They also included dimers, so every dimer that went through, and they also included GC content. And then they took the top 25% of guides, so across all cell lines, all genes, and separated them from everything else and said, these top 25% are the good ones and everything else is junk. Okay? So that's a little strict, but really what they're trying to do is they're trying to say, if you could have the perfect guide, what would it look like? And then they used a technique called machine learning to find these preferences. Machine learning is really just, you have all these features, and you try to figure out what are the different weights for different features to try to optimize your discrimination between the stuff that's good and the stuff that's bad. So you might say, here's a feature for T at this position, and G at this position, A at this position. Well, what does it matter, right? If I have a T at this position and it's not affecting anything, it's going to get a low weight. If I have a G at this position and it's making a big difference, it's going to get a big weight. Okay? So one of the main drawbacks with machine learning is that it's very hard to tell why results come out of machine learning. Machine learning crunches along and you get stuff and you look at it and you're like, well, okay, now I have a predictor, but it doesn't really tell me very much. But it can still be useful. So for example, from the machine learning, you can see, yeah, C, these now, this is the original data that you saw, and then these are some of the properties that came out of the learning compared back to that data. So you can see that, yeah, the machine learning picks up C is very bad, G is very good, but then there's, A is very good here, A is very good here. In the learning, A does not show up for these positions as being bad or good. It shows up as basically neutral. Here, C shows up as bad. Okay, that's fine. It turns out in the weights, G is assigned a worse weight than C at this position. So it's not totally clear why. This is one of the problems of machine learning. You can't go backwards and ask it, why did you choose that? Okay? But these properties do come out, and the good news is they seem to do pretty well. So in terms of back prediction, once you take sort of the entirety of the weights and put them together, you can get good discrimination between active guides and inactive guides. And very importantly, they took this same predictions and they compared it back to a set that they had never seen before. So they compared it back to the Lander-Sabatini screening data, and they were able to sort of post hoc pick which guides worked and which guides did not using this training set. But this is now something kind of important for machine learning. This becomes sort of a magic number set, right? You can't go in and change any of these numbers because these numbers work magically together. And you can't say like, well, I don't really like the weight that it picked for this. I don't think that's right. You can't go in and change that. It's going to change up the entire training set. So we've got a set of weights and we're going to use that. Okay? I think that's sort of one of the caveats. Machine learning works really great and training works really great, but it works on what you train it on. Okay? So when you're using this tool, really keep in mind what it is used for. This is used for knockout by a single indel. We're not deleting large sections. We're not doing anything like that. We're deleting genes. We're not doing CRISPRi. You would really want to hesitate before using this kind of data set on a CRISPR, designing CRISPRi guides, for example. These are used on mouse and human cell lines, trying to use the same guide rules in, for example, Drosophila or elegans. May work, may not. It's sort of unknown. It may be very different. And then finally, this is based on cell surface expression. So all of these guides are trained on cell surface expression, which may bias for certain compositions or may not. We don't know. It's really tough to tell. Remember, the protospacer is also sort of an underlying distribution of what the sequence itself is in that region. And so if they happen to, in some magical way, train for some certain type of indel, maybe a microhomology or something like that, that would be very difficult to see from the trained data. So I think this is a really great training set, and it's a fantastic tool because it seems to work very well. But really keep in mind when you use it what it's for. Okay? So it does find active guides very well. One thing that the Root Lab noted in their paper is that it can false report very good guides as bad. So they tried to take the very best guides. They do find that guides that work very well are reported as bad by their tool. But if a guide is reported as good, it seems to be good most of the time. One other sort of philosophical thing to keep in mind about this is it really assumes there is one true sequence for CRISPRs because they train based on sequence properties. Okay? So that means that in this model, when you train in that way, that suggests that there is a single sequence, like a magical sequence, that would be the best thing for CRISPR biology, or to introduce this kind of deletion. And that may be true or that may not be true. We don't, I would argue at this point, know really enough about Cas9 to know if it prefers one sequence or if it prefers some sort of more amorphous property of sequences that are not captured by this. For example, the propensity to form certain secondary structures was not trained on, and so that's not something that they would be able to pick up. Okay? This code for how they do this is available on the website. It does exactly what's advertised. I just want to point this out because this is now something very near and dear to my heart in terms of bioinformatics. There's been this really huge push in the bioinformatics community where if you make a tool public, if you do anything with bioinformatics, you should put your code out there because there are a lot of examples of people writing software and it doesn't do as advertised, or they make some change and suddenly everything's different. If it's not out there, you have no idea. Your results just change magically and you don't really know. So this code is available. If you like to program, you have someone in your lab that likes to program, someone like that, they could download it, make sure that it works, see how it works under the hood. Yep. So the question is the data generated from a lentiviral library? And yes, this is all lentiviral. So this is where you infect guides.
And it was just based off of the top 20 most active?
Yep, top 25%, where activity is defined as you have a surface expressed gene, and then you look for loss of that surface expressed gene. Then you sort based on now marker negative cells and sequence those guides from the lentiviral library.
And so using the results, are you able to take a look at the remaining 80 and see,
Yeah. Yeah, so that actually is what this means. False reporting good guides is bad. So actually, if you go back and you look at the other 75%, they assign very low scores to guides that are actually depleting. They are able to say, if a guide is really, really good, we will tell you it is really, really good. But if a guide is like sort of good, they're not able to discriminate sort of good guides from complete dog guides. Okay?
I haven't had the opportunity to do that.
Yeah.
You download it and try other algorithms.
So the question is, have we tried to retrain? No. So the code that they distribute on the website is just the code to generate the scores. I think they've deposited all of their sequencing data, so you could certainly go back and retrain if you wanted to. When I say the code's on the website, I mean like the code to generate the scores that they output, all of that is out. Yep.
Do you have any feeling for how other species might behave? I know you mentioned it's not really sure. Are people working towards that? Some are plant scientists.
Yeah.
I'm just wondering, where do we start?
Yeah, that's a great question. My hunch is that this stuff, unfortunately, might have to be figured out for each organism. Something that we have seen, certainly, is that even in human cells, different cell types will repair cuts in different ways. So we've got, the most obvious example in my mind is not to do with cutting, but with HDR. We have one cell line where you give it a donor template and we can get massive amounts of HDR. And then the exact same guide, the exact same locus, everything exactly the same, we get 0 HDR in a different cell type. So I think when you start moving to plants, it's going to be that rate much, much larger. So unfortunately, I think it's going to require more experiments. Think of that as an opportunity rather than something to paralyze. Okay, so this is what these look like. The tool is going to spit out protospacer sequences. It's going to tell you then how well it scores it. Here's just an example of a couple guides that it thinks are really good. You would just go through and you'd pick the top guides. Because it's doing weights, it just assigns arbitrary numbers to each thing. So it says, if I find a T at this position, +5. If I get a C at this position, -2, right? These are just magic numbers. They're on no scale. All they do is but this is actually where this code available on the website is. The program says all of these magic numbers. And so if you were to retrain on something else, you could easily just go in and plug your numbers into their script and run it. Okay, so that's talking about active cutting libraries. I'll go through this just very briefly because Max talked about this. Doing these kinds of tiling experiments is exactly what Max, Jonathan, and Luke have done for CRISPR-IA. So they just take genes of interest. Now instead of tiling over the gene, they tile around a region upstream of the gene and then look for things that will give you either inhibition. So this is what CRISPR-I activity looks like around a transcription start site, which is the dotted line, or activation. And again, these look inverted because you're looking for ricin phenotypes and depletion or enrichment of guides, but basically, it's really nice because you have a transcription start site and they basically mirror each other around that. So I'm not really going to get into details of what exactly the rules are because it turns out the rules are a little bit complex. They have to do with homopolymeric sequences and chromatin organization and that sort of stuff. But be on the lookout for this soon. And there will be a web tool incorporating this. Okay, so we talked a bit about activity of guides. Okay, we want guides to be very active, but it doesn't help us very much if we have a guide that is blazingly active, but yeah, it also cuts in 50 other genes somewhere in the genome. So we also want our guides to be very specific. So before I mentioned some of the things that just off the top of our heads we can incorporate. There are a couple other things for specificity that are just like that. We should probably avoid repeat sequences. If you look at a human genome, there are a whole bunch of sequences that are reported as repetitive, and these are very well understood, very well known, very well annotated in the human genome. Part of the reason they're so well annotated is that they're really a pain in the butt for sequencing approaches, and so sequencing has figured out what these things are because they're really hard to sequence. You wouldn't want to put a guide in them anyway, because if they occur thousands of times in the genome, you probably don't want your guide to be there. Also, something you'd want to avoid is targeting very conserved regions of a gene family. A great example of this for me is, so one of my favorite protein classes is deubiquitinases. I do a lot of work in ubiquitin signaling. And inside the deubiquitinases, there's a family of proteins called USP17. There are about 12 USP17s, and they have names USP17L1, L2, L3, L4, L5, et cetera, et cetera. And they're 99.99% identical on the base level, okay? So right off the top of your head, you can know that if I try to target one, I'm going to hit every single other one of them. Okay? This is sometimes hard to remember in general, if you've got a protein and you're interested in targeting it, it's not going to hurt you to blast it against the genome. We talked about BLAST as an alignment tool earlier. Just blast it against the genome. Find out what it matches to. And if it has completely identical matches to other parts of the genome that you're interested in, you probably shouldn't be choosing guides against that region. That will mostly come out in computational searches, though. So it's not something you have to spend too much time thinking about. OK, so now we get to more complicated things about specificity. And that's where we come to a server that probably most of the people in the audience have used. This is the CRISPR MIT server. This is one of the first specificity or guide design servers to come out. It's very frequently used. So I thought I'd go a little bit into detail about how it works under the hood, because since it was one of the first ones, I think most people sort of gravitate towards it. But most of the people that I talk to are not quite sure how it comes out with the scores that it comes out with. So I thought I'd talk about that a little bit. The data from this server is based on a really nice Nature Biotech paper from Feng's lab in 2013. OK, so if you wanted to find out specificity and you wanted to really design some way to look at specificity, I think there are two ways that you could do this. So you could either make one guide, and then you could say, what are all the places in the genome that hits? And since you guys have heard from Dana already about specificity, what does that sound like to you? What's a sequencing approach that you guys heard of where you make a guide and you find out all the places that it goes to? GuideSeq, exactly. So far, no one has gone back and updated any of these servers to incorporate GuideSeq results, but they certainly could. And certainly as we do more and more GuideSeq stuff, I'm sure we're going to see a lot of these tools, specificity prediction softwares, updated to include that sort of stuff. This paper is now a couple years old, and it was way at the beginning of CRISPR biology. So they instead took the approach to make a whole bunch of guides, at this point thinking, well, we don't know what all the possible off-targets can be, but we do know on-targets. So we're going to take one on-target that we know, and we're going to make every single possible variant of the protospacer targeting that guide. And then we're going to say, what of these variants still cut in this region? So for that, they chose EMX1. They chose a couple of different places in EMX1. And they made every single variant and asked which ones still target it. And from that, so I'm just going to show some of the overview figure from this paper. They turn it into this big heat map. Okay? So they look at all the different sites, and they make all of the different combinations of mutations. And they look at, well, what happens if G goes with T at this position, all that stuff. And then they collapse it all the way down into this one-dimensional map. Okay? And so here, if something is in a light color, that means that if you change that base, it doesn't target anymore. Okay? If something's in a dark color, it means that you can change the base, and it's still going to target somewhere. Okay? And based on this, there's sort of an arbitrary chain or an arbitrary line that people sometimes draw and divide protospacers into so-called seed and non-seed regions. Okay? So whenever you hear people talk about specific region of a guide, things like that, it generally comes from data like this. However, there are different ideas about where the seed versus non-seed actually is based on the data set. And so different people do different weighting schemes when you have mutations in one direction or the other, one region or the other. And so again, it becomes very important to look under the hood and understand, OK, which data set are they referring to? What is this tool actually doing? So when you use CRISPR MIT, it's based on this data set very, very strictly. And this is a seed region, and this is a non-seed region. Okay? So single mismatches in this region are not tolerated at all. Single mismatches can be tolerated in this region. It turns out you can actually have multiple mismatches. So if you have two mismatches together, you can still get some cutting if you have the mismatches somewhere back in the non-seed region. Okay? If you have 3 mismatches together or 5 mismatches, that seems to kill it. OK, so now we've come up with 2 seems to be OK, maybe in some places, 3 and up, no good. Well, those 2 mismatches, actually it matters how far apart they are as well as where the mismatches are. Okay? So if the mismatches are very far apart, it seems like that hurts more. There's sort of hurts less. There's sort of a caveat here, though, which is because of where the mismatches are put here, by definition, the other mismatch is in the non-seed region. And so some of this was taken into account in the training, but some of it is also sort of used in a very heuristic way, meaning arbitrary rules were set. Okay? And having more than 3 mismatches really totally kills activity. So how is this taken into account? So when I put up an equation like this, in general, people in the audience go, oh God, this is horrible. I'm never going to be able to understand this. But I'm going to walk you through it. And as soon as I do it, it's going to make total sense to you. Okay? So this is exactly the equation that this server uses. They trained the weights from the heat map. The training details are a little bit unclear. I'll put up the weights so you can at least see what it means. Okay? So here are the weights used along the sequence. So this is the first position. Now we're in the non-seed region. Then you get to the seed region. You can see the weights start to get higher. Okay? What this means is as you get higher and higher weights, it's worse and worse to have a mismatch in that region. Okay? Here, you can have mismatches. Doesn't really matter. Okay? What do these complicated terms mean? This is just the distance between mismatches. Okay? So if you've got mismatches close together, then this number is going to be very small, and you're going to start to get bigger penalties. Okay? This is now just penalizing total number of mismatches. So if this number is very, very big, then this term gets very, very small, and your entire score gets very small. So if you have a whole bunch of mismatches, the entire score drops like a rock. Okay? This is the term that most people freak out about. Usually it's because they haven't seen this symbol before. If people have ever seen the summation symbol, this is the exact same thing. It just means you multiply the terms together, okay? Instead of adding them together. So all this means is you just go through your sequence and you say, do I have a match or a mismatch, match or a mismatch, match or a mismatch, match or a mismatch? And if you do, you multiply those terms together. So if you have, here, you have an exact match. So you have a score of 1 minus penalty of nothing. So your score is 1. You have one mismatch here, which is this position. So you get 1 minus that times 1, 1, because you don't have any other mismatches. Okay? Again, similarly, if you have one mismatch here, you have this 0.685. That's this position. Okay? Every other term is 1 because you have no mismatches. So what does it look like if you have two mismatches? That means that you have 0.828 here, and this one is 0.685, so you just do 1 minus the weight times 1 minus the weight at the other position. That's all that this is doing, okay? And then you multiply by these mismatch terms. Is that clear to everyone? Okay? Yep.
This is for off-targeting as opposed to on-targeting.
That's correct. Yep. So this tells you nothing about on-targeting. So this would tell you how likely am I to hit some other position.
And those values are static at those positions.
Correct. That's right. That's right. So what you could do, though, is you could take this tool and then go into the root server and say, I've got this guide. It looks like it's OK in terms of off-targets. What is its on-target possibility? But at the moment, there does not exist a unified tool that will do both at once. OK, so if you guys have used the CRISPR server, you've seen that it outputs one score, right? But this is the score for every single possible match. So you would say, like, here's my exact on-target. Well, that's going to be a score of 1 no matter what. Well, what are the scores for this potential off-target or this potential off-target and this potential off-target? There could be hundreds of these. So how do you come up with a specificity score? All you really do is normalize to 100 and then add them together. I don't know why they chose those particular subscripts for this. I think it's probably some graduate student that snuck something by. In general, the way this works is you just say, I have a score, and if I have a whole bunch of off-targets, and those all get high scores, that's bad. Okay? If I have a bunch of off-targets and they all get low scores, that's probably not so bad. Okay? So it's just a summation. So if you have a lot of high-scoring off-targets, it's going to decrease the score. However, I'd really note that off-targets and exons are noted in this tool, and you can see them. They're really nicely shown. There's no penalty applied based on those. Okay? So if you have an off-target that happens in a gene desert, that's weighted the exact same as an off-target in the middle of P53. Okay? It's up to you as the user to know how that works, and that's totally fine. You just have to be very careful about that stuff, because even if you have an off-target that's way down at the bottom of the list, but it's in some gene highly related to the biological process that you're interested in, you'd probably want to avoid that guide, because there's a vanishingly small chance that you could get some sort of weird effect. Okay? The other thing I want to point out is that, so this uses the UCSC server. I talked to you guys about how you choose off-targets and how those are called. So this uses the UCSC set of genes. It calls those genes based on exons. So if you have a very high-scoring guide and it happens to lie in an intron, near an intron-exon boundary, this tool will not call that as a gene hit. Okay? It will only call exons as in-gene hits. So you should be careful about that based on what you're trying to do. Okay? So I think this is one of those cases where it's buyer beware, and users should kind of know what they're doing. The system, I'm going to very briefly just sort of show you guys, in case you haven't seen them, what they look like. By now, there's a lot of different species that get put into these. So for example, if you're interested in plants, I think Arabidopsis might be in here. Mosquitoes are certainly in here. A bunch of, I guess plants are not in this one. They might be in Chop Chop. Arabidopsis is? OK, great. I still can't see it. I'm blind. Oh, there it is. Great. So this is good. All this does, it doesn't change the scoring in any way. All it does is it says, when I want to call something in a gene or not, how do I know what's a gene? Okay? The output from this is really nice. It shows you where your guides are going to map against the sequence that you put in. It shows you all of these potential guides, and then every time you click on a guide, it shows you all of the potential off-targets. It's not totally clear where these off-targets for scoring are coming from. I mentioned that bow tie is used for some servers. It's not clear where these are coming from. Basically, how do you know whether you might want to score against something or not? But as you can see, these are pretty close to your on-target, and so it's figuring them out from somewhere. Other servers that are out there that might be really useful, there's one called Chop Chop. This is from Harvard. Chop Chop uses a non-trained scoring scheme. So it uses a heuristic, we would call it a heuristic scoring algorithm. This is another word for saying it's arbitrary. This is the scoring scheme. If you have 0 mismatches, you get a score of 1,000. If you have 1 mismatch, you get a score of 100, right? You just sort of assign arbitrary numbers. That doesn't mean that it's necessarily worse. For example, if you wanted to say, hits inside a gene of interest are really, really bad, add huge amount to that score, you could totally do that. That's no problem. There are times when you're trying to learn very complex features that machine learning and these sorts of trained algorithms can be very useful. But there are other times when you know obvious stuff. And this is something in general for you guys to internalize as an audience. If you know something, never trust the computer over what you know, right? The computer has no idea what experiment you're trying to do. It has no idea what your biology you're looking at. You should always trust your biological knowledge over a computer that says, this is a good guide, use this one, right? Do what you think is going to work and what you think is going to be best. So if you see some guide that is predicted to be super active and have 0 off targets, but you know from something that you've done in the past that, oh, this region is, in this particular cell line, there's a SNP that makes it similar to this, obviously you wouldn't take that. Yeah?
If these scoring methods are heuristic in nature, are they at least linear in scale?
No. Yeah, so these are not really based as much on patterns in the guide. They're more based on mismatches, bad GCs, things like that. So these are nonlinear, and they're really trying to just get you to look at certain guides. In some ways, I think that's probably a little more honest, because really, at this stage in the game, we're not very far into bioinformatics for CRISPRs, or even just in bioinformatics for genome engineering in general. And so if someone were to point me at a server and say, I have one guide that scores 55 and another guide that scores 0, or that scores 50, right? I'm not really necessarily going to choose the 55 versus 50, right? It's going to be very tough to know that there's a difference between them. And I think approaches like this are really just trying to make that very in your face, right? This one gets a score of 1,000, this one gets a score of 10. And that tells us that this one is, it doesn't have a lot of problems. This one, I can find a lot of things wrong with this. Okay? So it's really not trying to quantify so much as it's trying to stratify. Some nice things about Chop Chop. On the CRISPR MIT site, you can only put in relatively short sequences. This server allows you to put in any length sequence, and that's nice because it allows you to design T71 primers. So you've probably heard about T71. It's how you look for indels. And there's always this question about, where do I put my primers for that so that I can see where the indels are? When you go into Chop Chop, first you choose the species, You get a bunch of guides that align to, say, the entire gene that you're interested in. And then when you click on any one of those guides, it'll say, oh, here are the primers that I would make to be able to look at the indel around that region. So it can be really useful. However, since it doesn't do these kinds of quantitative off-targets, sometimes it can be useful to, say, predict the same region in multiple servers and see how they agree with one another. That's kind of the stage that we're at in the field. There are a lot of servers. I'm not going to go through all of them. I think the slides are going to be distributed at some point. If not, people are certainly free to email me and get them. This is sort of a preliminary list of some of them. People have started making servers that are specific for specific organisms. This gets to your question. And they may be more accurate in those cases. So I'm going to take the last couple of minutes and just tell you a little bit about what we've been doing at the IGI for this. So we've been really interested in making a general tool. As you've been seeing from these servers, they're really aimed at one task. They're aimed at specificity or activity or CRISPRi or CRISPRa. And we've been trying to think about taking all of the data that's out there from all of these really great papers and turning them into one tool. And one of the things that we've been trying to focus on to do something new is on HDR, so homology-directed repair. So trying to go beyond just saying we want to knock out a gene, how do we, in a very fast way, design both guides and templates to be able to knock mutations in? Just to get everyone on the same page, the problem with doing HDR is that maybe you have a whole bunch of guides that are potentially in some sequence. And what you really want to do is you want to change this mutation to some other place. Well, the way that you would do this is you would have to be deleting PAMs to keep your guides from cutting that region. And when you start to think about, well, I got all these guides and I want to design all these PAM mutations, well, those PAM mutations, they have to be silent. They can't change the region of interest. And I want to do this across a lot of guides and I want to try a lot of things. This becomes a huge amount of mental gymnastics to do. So we've been trying to solve this in a relatively fast way. So really what we want to do is we want to have a target sequence. We want to get some sort of scored guides out of it. We want to be able to cut, inhibit, or activate. We want to get activity scoring and specificity scoring. And the way we're doing this is, we're just going to copy what people have done that works very well in the past. So we're going to use databases that are already out there for activity and specificity. But we're going to try to do this from multiple sources to try to come up with something that maybe incorporates multiple things. But for HDR, we want to do this in a way that makes sense for a researcher. So we want to start with protein-based coordinates. We want to say not, in base number 5,236,000 of this chromosome, we want to say we want to mutate residue 5 in this protein into this other thing. We want to be able to mutate, insert, and delete. We want to automatically find places where we could silently remove the PAM. And we want to take into account things that we're starting to learn about homology windows, and then use that to predict HR templates. So we're running a little bit low on time, so I won't give a demo of this software just to show you kind of what it generally looks like. Unfortunately, it's a command line only tool at the moment. We're working on a web interface for it. Really what you do is, you just give it some sequence file, your sequence of interest. You tell it that you want to give it some output, what organism you're in, and then you tell it how long you want your template to be. And N2 just means how many guides am I going to use. And the really nice thing about this tool is that it will output homology templates that are compatible with all of your guides. So if you tell it, I want an HR template that's compatible with my best 2 guides, it will give you an HR template with 2 PAM mutations for your best 2 guides. If you tell it 5, it'll mutate out the PAMs for all 5, as well as introduce you mutation. So the output looks like this. Just to give you an idea, some idea of the scale of what you can do. This is the start of the output file from a run in which we're mutating out the active site of every deubiquitinase. There are about 100 of these. The run takes, I think it took about a minute to do all 100 deubiquitinases. This is showing 2 guides each, 2 different protospacers, and then identical HDR templates for each. So the way I ran this is by saying, give me an HDR template that's going to be compatible with both of those guides. I could have also said, give me an HDR template that will only work with the guide that you're paired with. I could have also said, give me one that's compatible with 5 guides, et cetera. Okay? So this is really up to you. This is something where, like I said, we're working on turning this into a web tool. And hopefully sometime soon, we'll be able to share it with you. But it is available on the web right now if you're not afraid of code. It's on GitHub right now. You can totally download it. It's under active development. So do expect the code to change out from underneath you. But if you have ideas for improvements, we're absolutely willing to take people committing back to that code. But also, like I said, if you just want to use it and have it be on the web, it's going to be on the web soon. And then one last plug for Nick, who I know some of you have been seeing in the small group sessions. He's been working on a computational way to, in a sort of physics-inspired way, not do machine learning of, well, there's a magic position at this base. I think you might have heard from Jennifer about the way that Cas9 does searches on templates. And Nick is working on ways to incorporate what we now know about the physics of Cas9 searches into a unified model that can predict both on-target activity, off-target activity, inhibition, activation, and cutting simultaneously. So the idea is, rather than having some sort of magic numbers that you train for one activity or the other, you instead learn something about the physics of how Cas9 works, and then you incorporate that into a general model to predict its activity. And why do we need these kinds of things? I just want to point out, this is, I think, one of the reasons why people in the room are here is because you guys are excited to be working with this. Well, it's an emerging field. When Keith did the GuideSeq work, he compared it to a variety of the off-target prediction software servers that were out there. And there's some overlap, but there's a lot of things that get missed. So it's sort of a little bit of a Debbie Downer note, but I do want to inject some sort of realism into this. Servers are just servers, and they can give you sort of danger signs, but you should never trust them with your life. They can show you things that are potentially dangerous. You should always be willing to expect that unexpected things are going to happen, and that's totally to be expected at this point in the field. Cas9 genome engineering is really in its infancy. And so I think there's a lot in the next couple of years that we're going to learn. There's plenty of room for improvement. One of the things that Jonathan and I were actually just talking about at the break was sort of a database that between my lab, his lab, and other labs in the Bay Area, we just collate all of the guides that have ever worked and all the guides that have never not worked. And once we have those lists, we can just put that up on the web. And if someone says, oh, I want to delete this gene. Has anyone ever used a guide that has worked for that gene? They can just go look it up. So if something like that sounds interesting to you, that's the sort of thing that is driven really well by communities of people doing things. And so I guess that's another thing that I would leave you with is, it's a nascent field, but it's a really nice community. People talk to each other and people work together really well. So if you do see a tool like that coming up online somewhere, please contribute your guides back to it. And then finally, so I blog about a variety of things, Cas9 and genome engineering related. In case people want to know really nitty gritty about how do I design a guide, go ahead and read this post. Again, I'll put out the slides. You don't have to scribble down this entire URL. But this is really a step by step. Step 1, look for this. Step 2, look for this. So a very kind of fine-grained version of what I talked about. And then I've been told that I need to tell everyone about the group photo, which is happening immediately after this session. So stick around. And with that, I'll finish up and find out if anyone has additional questions.